important day. I want to just sort of lay the groundwork for the day and what we hope to accomplish, and make sure no one goes deaf with my... Anyway, I want to start off by just thanking key people in the administration within our broader team who've been working and who've been really terrific partners here. So first, you're going to hear from Dr. Alondra Nelson next. She and OSTP, the Office of Science and Technology Policy, has just really been a critical partner, because what we're talking about today is innovation. What we're talking about today is new science, and there, OSTP really within the White House office really is the place you turn to for that kind of work. So so Dr. Nelson's and her team has been really critical. You're also going to hear after that from Dr. Tony Fauci, who needs no introduction, and you can't be talking about infectious diseases and thinking about a response and thinking about vaccines without having Tony really be the kind of intellectual leader for that work. And he's been such an important partner to me in my 3.5 months in this job. Tony, thank you for your leadership to this country for many, many years. And you'll hear from Tony all the great work that's already happening. And obviously there's a huge team of people that are behind today's event. But I want to talk a bit about what brings us together. Why are we here? What are we trying to achieve? I think if we take a step back and think about vaccines. You know, I think of vaccines as truly a miracle of human ingenuity, because they are the ultimate creators of natural immunity. People often talk about natural immunity as somehow infection-induced immunity. But what vaccines do is they harness and enhance a person's own immune system, activating it and training it to handle a disease. And vaccines, taken from a public health point of view, have been arguably our most powerful tool for eradicating and fighting diseases over the last 2 centuries. It is possible, I would make the case that it is possible that vaccines have saved more lives in the last 200 years than possibly anything else. Maybe except sanitation. But when you find yourself comparing something to sanitation as a public health good, you're in a pretty elite league. It really does speak to how remarkable vaccines are, and it's hard to overstate their impact in this pandemic, if nothing else. Many of you, if you were a representative of the American people, 70% of you have been infected with COVID, as 70% of Americans have. A large chunk of the world has. But what we know is that if you were infected after you were vaccinated, your risk of serious illness and death is dramatically lower. Just in the United States, the best estimates are that vaccines have saved over 2 million lives, prevented 17 million hospitalizations, averted close to $1 trillion of healthcare spending. It's fantastic. And it begs the question, why are we here? Like, we have fantastic vaccines. Why do we need something better? And my answer to that question is that the vaccines we have are terrific, but we can do better than terrific. In fact, I believe we have to do better than terrific. So why do I believe we have to do better than terrific? Because while our current vaccines are life-saving, this virus continues to rapidly evolve. And that means we need to constantly update our vaccines. Now, that's a challenge biologically and from a manufacturing point of view. It's also a challenge socially and politically. What do I mean by that? Now, we can make biological changes, we can update our vaccines. Predicting exactly where the virus is going to go is hard. But so is asking everybody to get vaccinated 2 or 3 times a year. That is also a huge challenge. So we need vaccines that are durable. We need vaccines that are broader and offer longer-lasting protection. We need vaccines that can stand up to multiple variants. Ultimately, we need vaccines that can protect us no matter what Mother Nature throws at us. Second, as I mentioned, these vaccines are terrific at preventing serious illness and death. But what we have seen is, given the contagiousness and immune evasiveness of new variants, these vaccines struggle against preventing infection and transmission, certainly over the long run. And so, despite the fact that 2/3 of the world's population has been vaccinated, we still see lots of infections out there. And so in addition to more durable protection, we need vaccines that prevent infection. We need vaccines that prevent transmission. So let me be clear about this. The bar for a new generation of vaccines is extremely high because the current ones are terrific. They're incredibly effective at preventing serious illness. They're also exceedingly safe. We've given out literally billions and billions of doses of vaccines of various types, and from a safety point of view, it's hard to think of something else that has gotten that kind of widespread use that turns out to be as safe as these vaccines are. So today we are going to talk about the science. We're going to talk about the manufacturing challenges. But we also know that science and technology are incredibly important, but they are not enough. We're also going to talk about making sure that these vaccines are equitably distributed. We're going to talk about how do we make sure that we build confidence in vaccines so they don't just show up on your door in a health system that maybe has not always taken great care of you. There's a lot of work that needs to be done if we are going to elevate the game and build on the progress we have made. So that's the goal. If you're going to do something like that, how do you do it? You bring together, of course, leading scientists, and we have them. You also bring together people in academia, some of whom are scientists, some of whom are not. You also bring together the private sector, because a lot of this innovation is happening in the private sector. And increasingly, that line between private sector and science is getting blurred, because there's great work happening in the private sector. And you bring together people who are business leaders who can take great products and turn them into billions of products that actually get the world vaccinated and taken care of. So that's the goal for today. We are doing more than just talking about this. You'll hear from a lot of people. There's great work happening within the broader US government, certainly at NIH, BARDA, rest of HHS. And the bottom line is that the administration, the Biden administration, is committed to doing everything we can to usher in a new set of vaccines, a new set of innovations and technology, and the policies that need to go with it. All right, let me finish off by saying and emphasizing a couple of key points. First is that this is going to take, from an administration point of view, an all-of-government response. So it's not just what will the White House do or what will HHS do. It really is going to be looking across the US government and saying, what can we do together? Second, it's clearly going to require partnerships with, as I said, academia, private sector, other scientific organizations. Third, it's going to have a global lens on it as well. We obviously are focused on what do we do to protect the American people, but this is a global pandemic. Global pandemics require global thinking and a global response. And we do all of this with a clear sense of urgency and purpose. Because again, while the vaccines are terrific, hundreds of Americans, thousands of people around the world are still dying every day. And while there's a lot we can do to continue to work on that and prevent that, building a higher, sort of a new generation of vaccines will make an enormous difference in bringing this pandemic to an end. So with that, let me turn it over right now to my colleague and friend, Dr. Alondra Nelson, who's the acting director of the Office of Science and Technology Policy. And actually, before we do, let me I was going to do one more quick thing, Alondra, I apologize. Let me actually quickly run you through the day, and then I'll turn it over to Dr. Nelson. So we're going to start off, obviously, after I finish, you'll hear from Dr. Nelson. And then you will hear from Dr. Tony Fauci, who really doesn't need an introduction, talking about the work of NIH in this area. Then the first panel, which is going to be moderated by Don O'Connell, who's our ASPR, for those of you not in government speak, is the Assistant Secretary for Preparedness and Response, focus on the next generation of vaccine design and manufacturing. We're going to have a little bit of a lunch break, come back. The second panel is going to be moderated by Dr. Francis Collins, who is the acting science advisor to the president, recently stepped down as director of NIH. And that's going to be on innovations in vaccine delivery. The third panel is going to be moderated by Dr. Lois Pace, who is the Assistant Secretary for Global Affairs at HHS. And that is going to be about innovations to advance vaccine equity, both domestically and globally. And then I'll come back and do some closing remarks. It's going to be a really great day. Thank you all for joining us in person. I know many of you traveled long distances. I'm grateful for your presence here. And for those of you joining us online, thank you for doing that. Now, over to my friend Alondra.
Thank you, Dr. Jha. Thank you for your convening us, for your leadership, for your partnership, and welcome to the White House, everyone, both here in the room and online. It's great to be with you all. As Dr. Jha mentioned, I have the great privilege of leading the White House Office of Science and Technology Policy, and our work really is to maximize the benefits of science and technology to advance health, prosperity, security, environmental quality, health, and justice for all of America. By founding mandate, OSTP's work is really focused on the future. We envision how science and technology can shape a safe, equitable, and flourishing world by tackling the tough challenges we face today, anticipating the unknown opportunities and obstacles that may lie ahead, and driving boldly toward both discovery and solutions. As we all know, we are still dealing with COVID-19 and its permutations in our lives, and we know there will be more pandemics, and we need to be ready for them. In a letter he issued just before taking office, President Biden asked, what can we learn from the pandemic about what is possible or what ought to be possible to address the widest range of needs related to our public health? And on his first day in office, the president issued 2 key executive orders, one on advancing equity and a second on strengthening the United States' response to COVID-19 and leading the world on global health and security. To answer the president's questions and follow his directives, we're taking steps to ensure that the United States is ready to respond effectively to future pandemics and to iterations of this pandemic. To do this, OSTP and our colleagues in the National Security Council, the Domestic Policy Council, and others have been carrying forward the work of what's called the American Pandemic Preparedness Plan. A key goal of this plan is to spur innovation in vaccine design, development, and manufacturing, preparing us for future COVID-19 variants and other infectious diseases. So now a short, but I hope instructive history lesson. The mRNA technology that led to 2 safe and effective COVID vaccines is technology that our U.S. Department of Defense kickstarted almost a decade ago through a public-private partnership to create a platform on which we could build a vaccine. So when the world shut down with COVID, we had a head start on a solution with the safest, fastest vaccine deployment in history. The world did this together in 314 days, and we're committed to cutting that time down to 100 days while maintaining our high quality and safety standards. The COVID vaccines that some of you in this room helped to create have saved countless lives. We thank you for your tireless, extraordinary efforts, but there is much more for us to do, and this is why Dr. Jha has convened us today. These vaccines lowered the death toll, reduced the burdens on our frontline healthcare staff, and allowed our children's education, our economy, and much of our lives to rebound. And it was federal scientific leadership and seeking future possibilities, taking risks, and investing in new technologies that made this possible. Now we and the Biden-Harris administration are working closely with global partners to accelerate development of broadly protective next-generation COVID vaccines. With the U.S. Agency for International Development and the Coalition for Epidemic Preparedness Innovations, we're working to develop vaccines that can thwart pandemic pathogens and save lives around the world. With the National Institutes of Health and BARDA, the Biomedical Advanced Research and Development Authority, we're supporting new private sector innovations like nasal sprays and skin patches instead of needles to administer vaccines in a more comfortable and accessible way, so that everyone in America and around the world can readily benefit from them. And across the federal government, we're doing much more to help protect people from COVID-19 and pandemic threats. We're working with the Environmental Protection Agency to promote ventilation and filtration that reduces the risk of transmission through indoor air. And some of you might have noted that in the room today, we have air filtration systems doing just that. And we're working with the CDC on its new Center for Forecasting and Outbreak Analytics to bolster the potential to track wastewater and other indicators to better predict, monitor, and address infectious disease and other public health threats. As Dr. Jha said, this is a whole-of-government effort. But there's much more to do, and that's why today we'll hear from transformative companies about how federal funding has helped them accelerate development of life-saving vaccines and creatively solve problems. such as figuring out how to store and ship vaccines without needing refrigeration so they remain shelf-stable and effective. And we know that developing safe and effective vaccines is only the beginning. We cannot fall back on the mentality of if we build it, they will come. People around the world still face a range of barriers to getting these vaccines. For people living in the world's most remote and least resourced areas, it's a challenge of availability and equitable access. For others, it may be the understandable challenge of a history of exclusion and mistrust. And for others, it may be the challenge of being subjected to a flood of inaccurate information. These inequities will persist beyond COVID and in future pandemics unless we adequately prepare and prioritize universal access to the innovations we're designing, developing, and producing. We need to make sure that the next generation of COVID vaccines is accessible to everyone. That won't be easy, but we must keep at it because new vaccine technologies themselves won't diminish people's hesitation to get vaccinated. We've done a great job of following the science in this administration. At the same time, we must also follow the social science, the behavioral science, and adopt more evidence-based public health communication tools as well. We need to use every tool in our toolbox. We can create a better future where we can develop effective vaccines within weeks of identifying a pathogen. It will be hard, but we can do it, where we support distributed manufacturing around the world, and where every country has the resources to manufacture thousands of doses in only a few days. That's the future that we're working to build. Oh, Tony, I'm advancing your slides here. Let me go back. To get there, particularly this part, I'm going to be really great on, I can assure you. That's the future we're all working to build. To get there, we have to continue to innovate, be creative, and propel great ideas forward into action. And we must continue to fund those ideas with support from Congress. We at OSTP are here to marshal resources across the federal government to improve health outcomes. And this fall, we will release the first annual progress report from the American Pandemic Preparedness Plan. This report will determine our next steps for implementing system-wide changes, including closing science and technology gaps, addressing vulnerabilities, preparing for emerging pandemic threats, and enabling the type of innovation that we're going to hear about today. This work really at heart is about taking care of one another, our loved ones, our families and neighbors, our fellow community, and the global society. And it's going to take all of us working together to achieve some of the audacious vision and goals that you'll hear about today. We need to transform our time horizons from years to days, just like we did with those first COVID vaccines, and we don't have a moment to waste. So now I want to pass things over with great honor and pride to Dr. Anthony Fauci, Director of the National Institute of Allergy and Infectious Diseases and Chief Medical Advisor to the President. Tony?
Thank you very much, Alondra, and thank you, Ashish, for the opportunity to make some brief remarks here to introduce to the group who may not fully appreciate, but I think many of you do, the role of the NIH in what Dr. Jha was talking about as being a whole of government response. We're responsible for the funding, many people I see in this room who we have, for the fundamental basic clinical and applied research, but we certainly cannot do it alone. And that gets to the continuum that Ashish was talking about, because we can bring the fundamental basic research concept and even bring it up to an early phase 1 or even further, but then we partner with other elements of the government and with industry. And that is, for example, through barter, through advanced development, which is a critical part of what we do, and then the all-important engagement of industry and other partners, such as CEPI and others. So this is not only an all-of-government response, it's really an international effort, which is really important. So I'm going to spend just the next few minutes in a very sketchy way, because I don't want to get into too many of the details, because this is going to be discussed by people who I do see in the audience. And that is, what is the role of NIH and why do we get involved in this? This slide is really an evergreen slide. I show it no matter what disease we're dealing with. What prompts us to use the talents of our investigators throughout the country and the world is that for us, we do research in immunology, microbiology, and infectious diseases for all of the areas you see there. And the two things that drive us are the public health need and the scientific opportunities. And where there are not scientific opportunities, when you have a brand new disease, you create the scientific opportunities. So very quickly, let's take a look at the public health need. We don't need to spend a lot of time on that. We really do know what the need is. A historic pandemic, the likes of which we have not seen in well over 100 years. So part one of the two pillars that drive us is clearly accounted for. Then you talk about the scientific opportunities. If you look at the results of what we have now as a result of science, this is just the vaccines that have now been utilized predominantly in the United States, but also worldwide. If you look at the platform technology on the left, the immunogen design with people like Barney Graham, who is actually in the audience here, that takes the fundamental science that has been invested for decades. And I think what people don't understand is that when you get started on a project, you don't start the day you get your first case of HIV or your first case of COVID. It's literally decades of investment. And that's the reason why we've had such success with the vaccines that was outlined by Ashish and by Alondra. And you know the numbers, Ashish mentioned them. We've already averted well over 2 million deaths, approximately $1 trillion in healthcare costs, and tens of millions of infections, as well as close to 20 million hospitalizations. That's the good news. What's the sobering news? The sobering news is why we're here today, because our job is not done. Because over the past 20 years, there have been 3 coronavirus outbreaks: SARS, MERS, and now COVID-19. Since September of 2020, this virus has continued to prove more than a formidable foe, with variants of concerns emerging, each with a bit of enhanced transmissibility. How do we know that? It keeps bumping off the prior variant, as we're starting to see now with the highly transmissible BA.5. Now, innovative approaches are clearly needed to induce broad and durable protection against coronaviruses known and unknown, hence what we're talking about, next generation. But there are 2 pillars of the next generation that we're all interested, concerned with, and pushing for. One is a pan-coronavirus vaccine that has both breadth and durability. That's important. And next, mucosal vaccine. So let's take a look at the pan-coronavirus vaccine. In December of 2021, my colleagues and I wrote in the New England Journal of Medicine back then that there was an urgent need for a universal coronavirus vaccine. Now, here's where we got to be a little careful, because if you look at coronaviruses in general, that is very aspirational. So what are we doing? We're going step by step. And you do that by looking at the coronavirus phylogenetic tree, as you see here. Now, the 4 coronaviruses that go from the alpha and then in the lower part of the beta are the common cold coronaviruses. And the 3 that you see within the beta are the ones we've had experience with since 2002. Namely SARS-CoV-1, MERS, and SARS-CoV-2. Now, another subtle concept that I want to make sure we discuss during this summit is that when you're talking about the ability to protect, it's very interesting, because what we were learning and are learning now about the common cold coronavirus may show some insight. A paper that came out literally a couple of days ago from one of our grantees, Alex Sette, showed that it's very interesting. Namely, you could have immunological memory from a virus that's different than the virus you're dealing now that doesn't protect against infection, but protects against severe disease. So now the pillars are dividing. You could say you may have protection against infection or not versus protection against severe disease. And we have to take that into consideration in our design. So, what about a pan? Let's start off simple. Instead of the whole phylogenetic tree, let's look at pan-SARS-CoV-2. We have multiple variants of concern that we need to deal with. And right now, it's even getting more complicating, because if you look at the SARS-CoV-2 family tree, we now have sublineages of sublineages, each of which has a transmission advantage for a number of reasons. One, it eludes protection from infection and prior vaccination, and vaccine-induced immunity and infection-induced immunity wanes. So you have two things going against us, which is the reason why when you look at what we can do, we've got to look at breadth and we've got to look at durability. That's a scientific challenge. That's not an implementation challenge right now. And that's the reason why you have to have science together with risk type of implementation, which was actually the formula for the success that we had originally when we were dealing at a time when we had no idea we would have these variants. That has changed, and we've got to be flexible enough to move with that change. So let's go to the next step. The pan-Sebecoviruses, namely one that would not only be good against the variants of SARS-CoV-2, but others might emerge. And the reason it's important is if you look right north of SARS-CoV-2, you see a number of the bat coronaviruses, which the most recent studies now about how you get evolution from a bat to an intermediate host to the human, it's very important for us to understand how we can get immunity that's vaccine-induced against this. So what is the NIH doing? Let's talk a little bit about it. Again, getting back to what I said, our job is not to develop a vaccine. We are not a vaccine production company. Our job is to facilitate by providing our scientists, brilliant scientists, many of whom are in this room right now, with the capability and the support to pursue the scientific leads, but also, and this is important, and I'm sure we'll hear about it later in the day, to remove any obstacles that are in the way of a very expeditious pursuit of your scientific goals. And we know there's a lot of them. There's enough animals. Is there enough this? There is enough that. Those are the kind of things that we can do. So since the start of the COVID-19 pandemic, NIAID has spent about $3.5 billion on coronavirus research, about $1.5 billion on vaccine research. And recently, we awarded about $43 million over 3 to 4 years at academic institutions, some of which are represented here, for the development of broadly protective coronavirus vaccines, and we anticipate additional awards within weeks. Now, to let the public know, to avoid confusion, when we talk about the development of a vaccine, I want to reemphasize, we can't do it without industry. So industrial partnership from the beginning and de-risking, as we have done rather successfully, is critically important. So when we talk about vaccines that are out there, almost invariable, I can't think of any that don't have fundamental basic science supported by the NIH with their fingerprints on that. We didn't develop it, we're not a drug company, but the fingerprints are there. And here are just some of the examples of broadly protective vaccine candidates that receive NIAID support. You're going to hear from many of these people. Some of them are already in various stages of development. Now, one concept that I think is important for people to understand, the key approach to broadly protective with breadth is you either take a common element of a virus and you then vaccinate someone that covers all of the subgroups. That's what's trying to be done with influenza. I'm sure many of you are aware of the stem approach because that's the least part of the virus that mutates. But another really exciting is a different approach that may get by without mRNA or be part of an mRNA, and that is a Mosaic approach in which you take multiple parts of the virus, multiple respective binding domains of different viruses, and you put it on a nanoparticle. And in fact, a paper just came out in Science 4 days ago from Pam Bjorkman and colleagues that did just that. And those are the kind of things that are the exciting science. This is just a few of the publications. I mean, I would have to just give you 5 slides to show them all. I've just taken some representative ones. So how are we going to enhance that? We have now awarded PO1s, or program project grants, to design and development of pan-beta coronavirus vaccines. And you see here it's Duke, Wisconsin, Rockefeller, Brigham, and many of the people who we're going to be talking about the basic science are involved in this. And then there's the mucosal vaccine. And again, the importance of mucosal vaccines. So I try to make this slide. This is my 5th version of the slide. I didn't want to make it too simple that you'll say, duh, and I didn't want to make it too complicated that the general audience wouldn't appreciate it. When you give an intramuscular or an intradermal vaccine, you get good systemic protection. You can get high levels of antibody that will spill over into the upper mucosal area, and for a period of time will protect you. But to ask to have astronomically high levels of antibodies that spill over is a non-starter, because if we did that, everybody would be hypergamma-globulinic with every stimulus you get. So you get good protection systemically, as shown with the protection against the lung, among other organs, depending on the virus. When you have a mucosal, and in this case intranasal, what you do is you stimulate at the local level, so that the goal of that is not only to protect against disease, but to protect against acquisition, and by acquisition, transmission. And that's really the holy grail of the 2 pillars: durability, breadth, acquisition, transmission. That's what we're talking about today. So the transformational promise of this, faster immunity recall, so that if you get exposed to the virus, you hit it right where it enters. Infection and transmission blocking, and needle-free delivery. And again, no need to go over all of these. These are just some of the candidates that are in various stages: basic, preclinical, clinical. One example of something that's very close to what we're doing right on campus at the NIH is that Jeffrey Taubenberger and his colleagues are using now an intranasal mist of inactivated whole virus vaccine, which is aimed at getting broad protection against both human and beta coronaviruses. Okay, so what are the challenges ahead as we come to the close of this discussion on my part? Looking forward to the follow-ups. 1, we need better validated animal models and methods to sample and quantify mucosal immune response. We lack really good correlates of protection. The potential delivery device considerations we need to expand. The theoretical safety concerns of some platforms, sticking a receptor binding domain in someone's nose, there could be a safety issue there. Unknown clinical trial designs and endpoints, regulatory pathways that need to be clarified, requirement, very hard and very important requirement for industrial partners. So what are the lessons we've learned from COVID-19 that we must apply looking forward? Consistent investments in basic science that leads to highly effective vaccine platforms and immunogens. We can't do concept development and early concept proving without fundamental basic research. We need to continue leveraging the resources that we have. One of the successes of the COVID trials was we leveraged the clinical trial network that we built over decades for HIV. We need to do that with these vaccines. Early partnership with industry and biotech in advanced development and manufacturing prior to the proof of efficacy, referred to as at risk. That led to unprecedented time frame for the availability of safe and effective vaccines, and finally ending with something we all know. The cost of major investment of research pales in comparison to the cost of a pandemic. Thank you.
All right. Fantastic. What a great way to get our morning started. Now I'm going to turn it over to Dawn O'Connell, who is our Assistant Secretary for Preparedness and Response, and what do you need? And she will introduce our first panel. Dawn, thank you.
Good to see everybody. Thank you for bearing with us as we get situated here. We've got an esteemed panel. Let's get started. So to all of you, thank you so much for coming. To those of you that are watching the live stream, welcome. Delighted to have you with us. So my name, as they just mentioned, is Dawn O'Connell. I'm the Assistant Secretary for Preparedness and Response at HHS. And I'm delighted to be here to moderate a discussion with our panel on the importance of next-generation vaccine manufacturing and design. And this is a topic of great importance to those of us at ASPR. Some of you may know that ASPR is home to the Biomedical Advanced Research and Development Authority, or BARDA, which supports the development, as Tony just said, of vaccines, treatments, and diagnostics to respond to public health emergencies. ASPR is also home, as of January 2022, to the HHS Coordination, Operations, and Response Element, or HCOR. That leads the coordination of the acquisition and distribution of COVID-19 vaccines and therapeutics. This is actually the successor to the partnership between BARDA, HHS, and DoD that was Operation Warp Speed. So together with our partners, we have delivered more than 785 million COVID-19 vaccines, products that have saved millions of lives and prevented countless severe illnesses. As impactful as that work has been to get to this point, we know that this virus is dynamic and continues to change. We heard that multiple times this morning. And we don't believe that we will be able to fully manage this virus without the next generation of vaccines, which could provide, as we've heard this morning, longer, more durable protection against a range of variants, both known and unknown. So we are here to explore how we as government and industry can work together to advance next-generation vaccines that are more durable, longer lasting, and allow us to keep pace with this ever-changing virus. So with that, I'd like to briefly introduce our panelists, and then we'll dive right into the discussion. So I'd like to welcome Paul Burton, Chief Medical Officer at Moderna, Mei Mei Hu, Chief Executive Officer, Vaccinity, Angela Huang, President of the Biopharmaceuticals Group at Pfizer, Sandeep Reddy, Chief Medical Officer, Immunity Bio, and Dr. David Kessler, Chief Science Officer for the COVID-19 response. So welcome all panelists. We are so lucky to have this esteemed panel here, and let's pull them into this conversation. So Mei Mei and Sandeep, this first question is going to be for you. We have developed safe and effective vaccines, as we've heard multiple times this morning, that have saved countless lives. The pandemic continues to pose challenges as new variants emerge. We're all experiencing that. And while our vaccines continue to prevent severe disease, hospitalization, and death, there is a need to develop, as we've talked about, next-generation vaccines that provide longer duration of protection and broader immune protection. Okay, so could you provide your perspectives on the challenges and how your technology might be able to address these challenges. May, let's start with you. What are your perspectives on the challenges we face, and how will your technology bring us through and address these challenges?
There we go. So first of all, I'm humbled and extremely feeling privileged to be here sharing the stage with innovators and leaders who've responded to the pandemic so rapidly. So congratulations. I'm a mom, right? I have 3 young children, the youngest of which is an infant. And so I think about what type of vaccine I want to give my kids, my loved ones. And, that means one that works, right? That has broad coverage, and that's durable, there's long-lasting effectiveness, but also ones that don't make you sick, and ones that are accessible to everyone that needs them. Because there's nothing more heart-wrenching as a parent than knowing that there's effective medicine out there that you can't get your kids, or that you don't have a choice to give your kids. So, our synthetic peptide vaccine addresses some of these in the sense that it's a multi-tope, right? It's a multi-tope vaccine that's focused on RBD, but as Dr. Fauci was saying before, it has multi-antigens there. And so we don't just go after the spike protein, but we also take important epitopes on the M&N proteins also, often in very conserved regions. So it seems in our data that we are able to generate longer-lasting half-life antibodies, potentially 2 or 3 times higher than the existing mRNA, and even more against the inactivated. We've been tested against multiple variants and are very pleased with the results, and extremely well tolerated. So virtually no systemic AEs like flu-like symptoms. And it's scalable and distributable, which is important because it's not just us in the U.S. fighting this. It's a global issue. So, from our perspective, the technology, the data is the one that's going to lead the way. And yet, so we haven't ourselves taken money from government yet, but I will tell you that public-private partnerships will dramatically accelerate and amplify our impact. And so I think it's important that we do so in a fiscally responsible way. And to echo others, in the first go-round of Operation Warp Speed, I think the government spent about $5 billion on vaccine R&D. So let's put that in perspective, right? $5 billion is about 0.1% of our pre-pandemic budget, 0.1%, or about 2 weeks of our time in Afghanistan. Or if you take the median family income in the U.S., 0.1% is about $80. That's $80 to prevent 2 million deaths in the U.S., 20 million globally. That's equivalent of $80 to, prevent 2.3 trillion in lockdown costs, or a 500-time return on money. I mean, who doesn't want a 500 times return on money? Not to mention that when you invest in those, Dawn, you actually bring brand new technology platforms to the forefront. So when we think about the mission to space, we had countless technologies come out of that, including, Teflon, cordless devices, water purification devices. And so I think it's important. And, we at Vaccinity, we're not just about COVID. We're about bringing the efficiency of vaccines to other chronic diseases. And anyway, I think it may have gone too long. But I think there are many challenges, but we are all equipped here in this room to take them on with the right resources and support.
May, thank you for that. And I think it's a critical point that you raise about the introduction of new platforms, things that we might use for this response or things that we might be able to use for the next response. So it's really an important service that this innovation brings to the future and what's around the corner for us. Sandeep, want to turn it to you. So tell us a little bit about your technology and how you think it might overcome some of these challenges we're currently facing.
Sure thing. Thank you, and thanks for inviting me to be here. It's very exciting, and I think it's an important topic, very timely. Basically, my entire thought process, I think, echoes what Dr. Fauci has already talked about. So we have 3 separate platforms that we're developing, all of which follow along, I think, the same lines of logic. So the first one is that we need to have a universal vaccine platform, ultimately to get buy-in from the populace. There's been a lot of, I think, misinformation, and so to win back that, to be able to argue, here's a vaccine that you can take today that's going to give you long-term durable immunity, that's critical. But in order to do that, we need something that has broad applicability. So one of the strategies that we've taken is to use more than just an S target. So going beyond even just the S and RBD, but looking at actually N. So the nucleocapsid is relatively stable compared to the S target in terms of mutational frequency, and is well conserved across multiple different coronaviruses. So with our adenovirus platform, we have an S plus N technology, and that's currently entering phase 3 in South Africa. And we chose to go to South Africa because it's sort of, the front lines in the battle, as it were, where there's emerging variants. And sadly, there's still a very significant unvaccinated percentage of the population. The second thing that we think is critical is to engage the T cell. And some of it is because our background, ImmuniBio, is really a cancer vaccine company, and we started as a cancer vaccine company. My disclosure is that I'm an oncologist, not an infectious disease physician. And so, coming from that perspective, one of the first meetings I was in when we talked about coronavirus was I was questioning, why do we need antibodies? And they wanted to kick me out of the room. But, at the end of the day, the truth is that we need both. Clearly, we need antibodies, but we also need T cells. In order to break the cycle of transmission, T cells will be necessary to clear virus, and we think that that's critical. And I think the last piece that's important that we've learned across the industry is the value of adjuvants. So if we could take a very low-cost, simple, easy-to-manufacture protein-based product, but combine that with an adjuvant that could give us that T-cell boost and give us that pan-immunity, I think we have something that at scale, from a cost of goods perspective, could be used universally. So these are three of the platforms we're looking at. So our RBD platform is actually the one that's now approved in India, but with a different adjuvant, and we're looking at an alternative adjuvant to combine with that to get an even greater T-cell boost. So we've taken perhaps a slightly schizophrenic or shotgun approach, because I'm not sure we know today which one is the winner or whether there is a winner. And I think to Dr. Fauci's slide, you have multiple different approaches that are in play, hopefully all of which will succeed, because I think the more options we have, the better it will be for us in the future.
Thank you. We talk a lot about the benefits of having multiple shots on goal, to use one of our sports analogies. And so it's interesting to hear the different ways in which you guys are approaching that, Sandeep. So thank you for that. Let's turn to another question. This one will be for Angela and for David. The historic public-private partnerships we've talked about this morning a little bit already between the government and industry led to the development of first-generation vaccines on a timeline not previously seen. As we think about the development of the next generation vaccine and overcoming these challenges that we're talking about with this pandemic, the ever-changing variant compilations, what role do you see for the USG as we move forward? Angela, why don't we start with you, and then we'll turn to David.
Well, thank you for the question, and also thank you for having all of us here today. Pfizer has just been honored to be in this journey together with the U.S. government and with the world. And we are, just deeply committed to finding solutions and continuing to be part of the solutions for the world. We've learned a lot from the time that we've had over the last several years. And I want to say that fundamental to our ability to bring this vaccine forward was an incredible partnership with the U.S. government. But I would say that there are some areas of learnings that I think we can build off of. As we've talked about today, to be able to solve a problem this big, this massive, it really takes just a tremendous amount of stakeholders and a tremendous amount of commitment, investment. We talked about investments at risk today, incredible innovation. But I also think that there are other enablers that really, I think, came to the forefront. And first, I will point to what we began. Dr. Fauci began his talk today about the incredible science that exists and how that needs to lead the way. And in order for us to continue to do that, we have to ensure a thriving innovation ecosystem. That means that we need to have protection, intellectual property rights, intellectual property respect, so that scientists and industry can continue to do their good work. So that's fundamental, fundamental sort of like cost of entry into this. But I think we learned a lot as well from the time that we've had and how we were able to bring a vaccine into the market at such unprecedented speed. And first, I would say that there is a learning and a continued work that we can do together to ensure that there is a fit-for-purpose regulatory approval process, right? So we can have a great drug, a great therapy, but if there's no way to get it onto the market and improved quickly, that does not help patients and does not help the public. So I think the flexibility and the fit-for-purpose way of looking at what is the issue at hand and what is the kind of approval that we need, I think is one key enabler. Secondly, the clinical trial process and the actual conducting of the trials and the recruitment of the trials was another area where speed really helped us. And I want to really talk about the use of digital technologies and virtual tools that enabled us to recruit at incredible speeds, but also to recruit in diverse communities, because we wanted to make sure that at least 30% of our clinical trials for community was represented by minorities. And in this instance, we were able to use telehealth consults. We were able to deliver to our patients' homes the product, the clinical trial product. We were able to conduct virtual visits and follow-ups. And all of this meant that we were able to bring the trial to the patient rather than have the patient come to a site that may be too far. And that's how we were able to increase the number of patients that we had hoped to have rather quickly. In fact, in just over 100 days, we recruited 47,000 patients, which is sort of unprecedented in the short time that we had. But I think what really helped us there was the application of technology and the ability to bring the trial to the patient's home. So I think that would be another area that we can continue to partner on and to build off of. Importantly as well, I want to say that the kinds of trial design was another enabler of speed. And here I want to point to adaptive clinical trial design. Typically you would go through the phase 1, phase 2, phase 3, but in this moment we really didn't have the time to do that. So there's the incredible willingness of the FDA and other regulatory agencies around the world to partner with us and to really have robust conversations around what are the pre-specified endpoints and outcomes that we expect to see by when, and what are the safety measures and bar that we all want to aspire to be able to have that kind of discussion way up front and be able to then adapt our clinical trial designs according to that is what enabled us to get to the end quickly. So I think it would be great to be able to see the application of this kind of trial design beyond what we did just for the first COVID vaccine, but to other possible vaccines and to other possible therapies as well.
Angela, thank you for that. It is interesting. One of the lessons we don't want to lose in this response are those adaptations we made together, where we were able to adjust quickly to be able to provide this very safe vaccine through clinical trial adaptation and the other pieces that you mentioned. So we want to make sure that we don't lose sight of that as we head into the next generation. Very important work. Now, David Kessler, you've been part of the government's response for the last several years, so you've seen this firsthand. What would you say is the role of the U.S. government moving forward with the next generation work?
To date, because of the effort of all of you, the collective we, have delivered 784 million doses of vaccine safely. As of today, there 601 million doses have gone in arms. That is historic. Dawn, all of you, take a bow. I think that's the context. I think we recognize, I think we've heard this morning, I mean, there is a consensus that in light of all of that, that we need vaccines with longer duration and greater breadth of protection. I think we have that consensus. No one is, resting where we are. I mean, our colleagues at the VRC and in industry, certainly just intramurally, as Dr. Fauci has said, extramurally, our colleagues are working, now on intranasal delivery to better protect against transmission, nanoparticle vaccines that include multiple antigenic determinants. That is underway. There is, from all of that gone on, there is an incredible team. You live it, Dawn, your colleagues at ASPR, at BARDA, at FDA, at NIH, at CDC, at industry, at DOD, that are poised to overcome the challenges, develop the robust pipeline to support advanced development. I think we have to get these vaccines out there in the next generation, so we are exploring, the processes of going commercial, right, while still ensuring access for the un- and underinsured. It's a very important thing that we have to continue to achieve. I think bottom line, there is an exceptionally strong case, for investment, right? I think both, the Congress, through their wisdom in the past, the capital markets, industry, I think that continues and really is essential.
David, thank you, and thank you for those reflections on the work that we've done and the work that needs to come, and how energized the U.S. government is to see this work through. So, Paul, this question is for you. In the U.S. and globally, we've expanded manufacturing capacity to support global-scale manufacturing of COVID-19 vaccines, including critical components necessary for manufacturing. From an industry perspective, how do we maintain this capacity for current vaccines, next-generation vaccines, and for future pandemics?
That's a great question, and thank you, Secretary Connor. It's a privilege to be here to represent Moderna today. Look, I think distributed manufacturing, which is really decentralized manufacturing, is a key part of the future. For Moderna, we have announced recently collaborations with governments in Australia, the United Kingdom, Canada, Kenya as well, where we will actually be able to build plants, manufacturing facilities, R&D facilities in those countries, make product for that country, in that country, by that country. It's all part of an important ecosystem of health security, which I think many of the vaccines that we're fortunate enough to have today can bring to population. So that's very important. We certainly partner with manufacturing organizations, contract manufacturing organizations around the world. As vaccine technology continues to evolve, production of vaccines becomes more and more technically difficult. But the size of the facilities that we need to manufacture actually becomes smaller. mRNA is highly reproducible, can be made with high fidelity in very small manufacturing kits, but we can collaborate with those manufacturers. And also here in the United States, we've expanded manufacturing in our Norwood facility. So those are really the technical approaches that we're trying to bring for that decentralized manufacturing around the world. And I think the theme of all of the discussions is, we have to continue these discussions, continue these collaborations to instill confidence for people who at this critical juncture, critical part of the pandemic, have to have confidence, as Dr. Kessler said, 600 million doses in this country given alone. We know everything really there is to know about the safety profile of these amazing medicines. So getting that confidence and then the ability to get these shots made in countries and into people's arms, I think, is how we're going to continue to get out of the pandemic through this endemic phase and then into the next generation of manufacture.
And, you know, it's just an important point to talk about all you all have done globally to bring this forward and how important this capacity is for all parts of the world. And so, you know, the investments we're making now, we will have to leverage moving forward. And unfortunately, it looks like we're having multiple opportunities to do that. So thank you for that, Paul. Appreciate it. We have some questions from the audience, so I want to make sure that we get these in while we have the opportunity. Now, this question's a little provocative, but I'm going to go ahead and ask it, and then we'll kick it over to our panelists. And we don't have the benefit of FDA on this panel. We have the benefit of a former FDA commissioner, Dr. Kessler, but you don't need to speak on behalf of FDA, of course. The question is, how long will it take for these vaccines to get to licensure? Now, without having an FDA person on the panel to respond to exactly how FDA might approach this, what I'd encourage our panelists to talk about is the way in which you would accelerate getting your data forward, doing your clinical trials quickly, so FDA at the proper time would be able to evaluate it. So, Paul, why don't we start with you?
Yep, thanks, Dawn. I can say just a few comments. So I think, you know, transparency of data is fundamental to everything. Getting it out into publications, preprint servers today, describing it publicly is key. That gives people confidence, continues to give them confidence. You know, I would say that we were able to go from the recent VRBPAC meeting to actually manufacturing a vaccine today that contains BA.4/5, and that was, probably under a month ago. So I think we can manufacture very quickly. We can get into animal studies quite quickly. I would say the debate now that will go on in a august community like this and socially is, what degree then of animal data do we need versus human trial clinical data do we need? You know, that will certainly determine how quickly we can get to application and licensure. But throughout the whole pandemic, I think what we've been able to do is really partner with regulators around the world, certainly the FDA here in the US, and we've learned so much about rapidly but with great quality and certainly never sacrificing safety, how we really can speed these applications down to weeks.
Maybe, what is your perspective here?
So I think the theme of this summit is really about partnership, and it's about engaging early together with the regulatory authorities to understand what's needed, right? Especially in this changing dynamic. The situation is constantly evolving, and to be flexible and to be reactive and to be adaptive to that, I think, is incredibly important, especially for a small company like us, to know what does the agency want to see, what do they need to see, and to know that up front as best as we can. You know, LMICs are a core part of our strategy, and so we're working with multiple globally recognized regulators, both high income and low and middle income, on accelerated approval pathways. And, you know, I think engagement is the critical path, and being in front and having an open discourse. So that's at least our wish. And I think the FDA has done a fantastic job, obviously, since the beginning of 2020 in doing that. So I would hope that it would continue.
Great. Thank you, Mei. Angela, how would Pfizer approach this?
Well, I think the theme, you know, we've talked about here a lot already, and I just want to, you know, I agree and concur that engagement, constant dialogue, there's no substitute for that, right? An understanding of what are the agency's goals, what are the requirements, and then how can we meet them in a flexible and creative way, especially in a time of the pandemic. The only way you can do that is to do that, you know, constantly with each other. So I can't underscore the importance of a continued and excellent and collaborative, you know, relationship, which I do believe we've had, and is actually you know, we reap the success of that relationship that we've had, and the scientific exchange has been, you know, so vital. The other thing is what I mentioned, which is, as a company, what are the ways and how can we reimagine how work can be done to cut out white space and to be able to bring the same outcomes but in faster, right? So the application of technology, digital, you know, new protocols, the examples that I've used previously are the ways that we have and we will continue to innovate around to ensure that we can get out there faster and better. But maybe one thing we haven't talked about today is the importance of real-world evidence. We've just talked about how many people in America have had the vaccine, and actually around the world, probably 2.5 billion people have received the Pfizer vaccine. That's an incredible wealth of real-world evidence that we're sitting on. And so I think that we have a great opportunity, having that strong depth and breadth of data, to use that to also help us to understand and how can we design new therapies, but also how can we apply it to the public in a to facilitate, to expedite, and to be able to gain access quickly so that the public can really benefit from this.
Thank you. That's a really good point. Sandeep, how would you all approach this?
Yeah, I mean, I think, obviously I agree with my colleagues, but I think one of the most important and really prescient issues is identifying what would be appropriate correlates of protection that regulatory agencies would be willing to accept for approval. It's very clear that that actually is something that is at the forefront in other jurisdictions. So, for example, in India, there's a vaccine that was approved based literally on scientific data showing that it generates a sufficient immunologic response. And, when we're talking about shortening these time frames, I think the reality is in a world where, you guys can tell me how many 100 million people or billion people have been vaccinated, right? 2.5 billion at least. It's going to be very difficult to do a traditional endpoint type study. But it's very easy and very rapid to do a correlates of protection type study. And for us, I think we look at it not just in terms of humoral response, but also cellular mediated response. What is a sufficient and clear goal? What's the goalpost that we need to hit in industry? If that was known, that would make it very easy to design those trials. And I think going forward, that's going to be critical to get us to where we need to be. When we think about, and I'll just pick up one of the points that Paul made, when we think about, for example, self-amplifying RNA, from a manufacturing perspective, we don't need massive scale. We can do this, but what we don't have is unlimited number of patients. And so having some alternative way to be able to identify success and identify success rapidly, I think that's a very critical need at this juncture.
Thank you for that. David, any additional thoughts or comments?
Yeah, I am absolutely 100% confident to the leadership of FDA. I think we have seen they have risen to the occasion. They are creative. They are nimble when it comes to these vaccines. I have a great deal of confidence. I do think there are hard questions ahead of us, right? With the vaccines, with the effectiveness, the very high effectiveness against serious disease. What is, quote, better going forward? Is a hard question. I think we're getting closer on not necessarily a perfect correlate, but with correlates that will help us. I am concerned. I think one of the big impediments over the last several years is I think we have been, I am enormously grateful for our international colleagues, but for the Israeli data, but for the UK data, right? That has been essential. I think we are too reliant, on some of the international, they're great, but your point, Angela, on real-world evidence data, that very strong epidemiological data, I think, is absolutely critical, and we have a long way to go to develop those systems.
Very good points. Another question from the audience. One of the historical challenges for vaccines is the mismatch between the money needed to develop them and the ability of those who need them to pay. How do you foresee a path forward for public-private partnership that threads this needle? So we've talked already about the public-private partnerships and the need for that, but how do we manage this piece? Anyone want to try to take a first stab? Angela.
we've had tremendous learnings from the distribution of the Pfizer vaccine into the developing world. And if I think about the lessons learned, I think you can look at them sort of in sort of two chunks. The first is, how do you get the vaccine distributed there? And then secondly, you have to look at, how do you actually get the vaccine used? And I think on the first part, the ability to supply, we were able to meet that challenge, and we were able to meet that challenge really well. In fact, of all the vaccines that were distributed in the world in 2021, 1 billion of that went to the developing world. So I think in terms of equitable distribution and the ability for people to access it, they were able to do that. I think what we learned, unfortunately, is that the ability for these countries to utilize them was a different thing. And there are large issues related to vaccine confidence, misinformation, ability just to have, enough healthcare professionals administer the vaccine. And so if you look at sort of the last mile challenges and the complexity of what that takes for someone to actually get the vaccine, I think that we did not spend enough time there. And so one of the things that is going to be really important, well, has become important, but will continue to be important in order for everyone in the world to receive the vaccine, is healthcare system strengthening. It's everywhere. I would say that's in parts of our country, but also very, very, visibly in the developing world. And it's one of the reasons why, as a company, we have advanced on our thinking, and we are in an agreement and in partnership with several African countries, actually as part of a different initiative that we have launched called An Accord for the Healthier World, which goes beyond vaccines. It's about making all of Pfizer's innovative products available at a non-for-profit price to developing world and to the low-income countries. Part of that is not just about distributing and dropping the medicines off. A big part of that is about understanding what is needed for capacity building, healthcare system strengthening, and bringing together, talk about partners, more partners in these countries, so that we can really understand what it takes to be able to administer and receive the vaccine, not just supply it. So I think the supply part was something we understood really well. I think the remainder, last mile part is something that we need, I think, the entire world to focus more on.
Absolutely. Paul, did you want to jump in?
Just very quickly, Don. I know there's going to be a panel later on equity as well. Clearly, we partner with COVAX, CEPI, Gavi, other major institutions around the world. But I think just bringing it closer to home, Medicare will clearly take care of people over 65. There's the Vaccines for Children initiative for those younger kids. I think the people between 18 and 65, those underinsured, uninsured, is very important. And I would just call out the initiative of the President, the Vaccines for Adults initiative, which I think is extremely insightful, providing access there for vaccines that would be ACIP endorsed, I think is a tremendous step forward in access.
Absolutely. Thank you, Paul. May?
Yeah, so I think when problems are global, we have to proactively address them as global and not have the rest of the world as an afterthought, right? When you think of it as a global launch, I think you prepare and think about all the things that need to happen. Fortunately, vaccines are one of the most prolific medicines in our history, right? Over 80% of the people, even pre-COVID, were vaccinated, and because of that prolificness and that accessibility, they've been one of the most impactful medicines, probably doubling our life expectancy over the last 100+ years. I think technology really is an enabler there. So yes, we can find a vaccine that works, but can it be scalable? Can it be cost-effective enough that it can get to and can be affordable? And can it be distributable, right? So can you make it so that it doesn't require cold chain storage? Can you make it so that it can survive anywhere a Coke can go, right, to every part? And I think the technology is really has historically been our equalizer and our enabler, and that's where we should look at to solve these problems. technology has always equaled equity. Look at the printing press, right? Prior to the printing press, books were the most expensive commodity on earth. They were more expensive than gold by weight. It's because they had to be handmade, and monks had to literally write them. Then the printing press came around, and we democratized the book, right? I think that's where technology comes in. So how do we democratize vaccines? And I think we're on our way, but ultimately technology has always helped us make that exponential leap to create equity across the globe. So that's where I would focus on new.
Thank you, Mei. Sandeep, did you want to get into?
Yeah, I think so. I mean, first of all, I agree with what particularly what Mei just said. You know, I think it's interesting. So we've been fortunate enough to put together about 1 million square feet of manufacturing capacity without taking any public money. And the reason is that we started, again, as I mentioned earlier, as a cancer vaccine company, so we were able to pivot. And really the underlying point is that there is capacity. There's significant capacity. And the question is, what is it used for, right? So that's number one. Number two, the ability to produce vaccines at scale at low cost is not an issue, right? I mean, that's, as Mei pointed out, the majority of the world is vaccinated against a variety of different diseases. So we can do it. It's just, what is the right vaccine and how fast can we get it out there? Now, as we move into this next generation of vaccines, what's exciting is the ability to possibly lyophilize these and take us out of cold chain. I think, and I don't want to steal from the upcoming panels, but it'll be very interesting to hear about mucosal delivery. I think getting IgA and getting reduced transmissibility will be very exciting. If we could get into that type of environment where a lower dose can be used, right? A much lower dose. Now the scalability becomes very approachable, right? The COGS go down dramatically. If there's a several log reduction in the dose, there's a several log reduction in what we need to do to manufacture it. So the underlying point is that absolutely technology is here. I don't think it's coming. I think it's here. And we just have to apply that technology appropriately. So it'll be very interesting to hear about vaccine equity, but I think we have the solutions. We just have to implement them correctly.
Wonderful. David, did you want to comment on this?
Yeah. Can I, just to underscore a point that Dr. Fauci made earlier, the investment didn't begin for COVID in 2020. Investment began for COVID, right? You go back to Moderna years ago, BioNTech, Pfizer years ago, the NIH, a decade plus. That's what we built, the current response. That's what we have to recognize, right? And that investment in that basic research through the American people, through their Congress, right? You can't just pull a vaccine out today when you need it. You have to pull it, as my colleagues have taught me well, based on all that science that has gone in the years past. That's what the investment has to be.
So I'm going to do something unusual for our last question. We're down to our last 2 minutes. I'd like each panelist to tell me what you think the benefit of this next generation vaccine is going to be to the American people, to the world, if we can land it. What will it mean to people? People are exhausted. They're tired of COVID. They've been struggling with this for 2.5 years. If we can get this next generation vaccine, what will that mean? 1 or 2 words, 1 or 2 sentences, and we'll just keep moving down the line. Paul, start with you.
Well, look, I think it will give confidence, a way to finally leapfrog the virus, get out of the pandemic phase, truly into endemic phase, and cement a foundation for coming pandemics, which sadly, as Dr. Fauci and others said earlier, will doubtless be a part of the future.
Mei, what will this next generation vaccine mean to America and to the world?
I think it'll set an example to folks that we can do it and that we can tackle other diseases plaguing humanity, right? That we can tackle scientific problems together, that we can build trust with the public successfully, that we can save lives by working collaboratively, and that we can set up new platform technologies that can then be leveraged for other diseases. And like I said, we're working on COVID vaccine, but that investment is broader than that. It can, we're working on vaccinating for Alzheimer's, migraine, heart attack, and stroke. These things are not just singular in their impact. They build on each other, they're exponential, and I think that showing an example for that will mean a lot for everyone.
Angela, what will this next generation vaccine mean?
I think that the ability to create a next generation vaccine will just demonstrate once again that as a country, as an industry, and as a scientific community, we have the tools, we have the expertise and the ability to rise to the challenge, whatever that challenge is. And so that should create a great deal of confidence and security in who we are as a scientific community. I think importantly for all of us who are just citizens, I think just relief, healthy living, the ability to conduct life in the way that we want.
Sandeep, what will this mean?
Yeah, I mean, I think normalcy, whatever that means, right? Whatever that means. But also, I think ultimately cross-reactivity and memory from a vaccine will give us the confidence to, if not just this pandemic, but as Dr. Fauci mentioned, we've had 3 essentially in the last 20 years. But let's get rid of this pandemic, but the next one too, and the next one, and the next one. And if we have that ability to prevent, that's really what we want out of a vaccine is prevention.
David, what will this mean?
I think the current vaccines have shown that they can prevent. You don't have to die. You don't have to be seriously end up in an ICU. I think that's what the current vaccines have done. But we don't know, how this virus may continue to mutate and come back in a stronger form. And I think a next generation vaccine will safeguard and be able to allow us to avoid the death and disability from COVID in the long run.
Well, thank you. And I want to thank all of the panelists. You can tell how lucky we are to have such strong partnerships with innovative thinkers, people who are facing this complexity with vigor and creativity and, a desire to get us out of this pandemic and to help us get to this next generation vaccine. So I want to thank each of the panelists. I want to thank the audience for the questions that we received. It was terrific to hear from you, to know what was on your mind. And I think we're going to break now for lunch. And those of you that are live streaming, we'll be back at 1:15. Thank you, everybody.
talk about innovation in vaccine delivery. I'm being told I can't be heard because I'm worried about the microphone. Is it possible we could turn up the PA system? Just a question here. Yes, I'm getting their attention. This is good. So we have a distinguished panel here. And we're going to have fun talking about the science of vaccine delivery. I'm going to take off my mask now, and since we are supposedly going to be articulating in a particularly clear and coherent way. And I have with me people who are going to fill you in with some of the most interesting developments that are happening. In Tony Fauci's really remarkable summary this morning, he talked about two major ways in which we can see the future of vaccines for COVID evolving, one being the way in which one can come up with ways to immunize against common antigens or perhaps do the major multiplex Mosaic thing. But the other thing he mentioned was mucosal administration, which happens to be not the only way that we might come up with vaccine deliveries that are not the same as what we're all used to with needles into arms. And we are going to hear about that from this group, both in terms of nasal and also microneedles. And we also have distinguished basic science expertise about the immune system and particularly how it relates to this topic. And we're also going to have the chance to have a real-world estimate about how this would all be seen as a useful addition to the public's opportunities to take advantage of vaccines. So let me quickly introduce the speakers who are here in alphabetical order, and then I will ask each of them to speak for exactly 4 minutes, or less would be all right, and then we'll engage in a bit of a conversation about issues, and then we're ready also to take your questions. I've already gotten a couple of questions that have been submitted. Maybe there will be others in the course of the next little bit. So next to me is Dr. Miao He, who is the founder and chief executive officer of CyanVac LLC. Next to him, Dr. Akiko Iwasaki, who is the Sterling Professor of Immunology and Molecular, Cellular, and Developmental Biology at Yale University. Next, Marty Moore, who is founder and chief scientific officer of Nasal Vaccines. Next, Mark Prausnitz, chief scientific officer of Micron Biomedical. And finally, Michelle Smith, director of the Office of Health Equity and HIV Elimination, Arkansas Department of Health. I couldn't help but notice in looking at the bios that three of our members of the panel have connections with Georgia, and I don't know what to say about that. I just had to comment. University of Georgia, Emory, and Georgia Tech are all represented here in various ways. So something about their ability to do innovative things, or maybe having the CDC next door has something to do with it. Anyway, welcome to all of you. I'm going to go in a particular order because I think it might build this way from sort of the basic science to the public application. So first, I'm going to ask Dr. Iwasaki to give us her 4 minutes of what is most important to get this conversation going. And you may need to get close to the microphones. I noticed in the first panel that if people were a bit far back, it was hard to hear because I was sitting in the back row. Okay. So please proceed.
Hi, my name is Akiko Iwasaki. Can you hear me in the back? Okay, wonderful, thank you. So I'm a professor of immunobiology at the Yale University School of Medicine and an investigator at the Howard Hughes Medical Institute. I've spent most of my scientific career studying mucosal immunity and developing mucosal vaccines against viruses. I'm here today to talk about nasal vaccines to help end this pandemic. And as you know, SARS-CoV-2 is a virus that's transmitted in the air. We inhale these viral particles. They land on our nose, our mouth, our throat, and in the lung, and start the replication process. Currently, the conventional vaccines do not induce mucosal immunity, although it induces very robust systemic immune responses that circulate in the bloodstream. Okay. So decades of research have shown that mucosal immunity requires mucosal vaccination. So mucosal vaccines induce local immune responses that are compartmentalized and specialized, such as IgA that's secreted into the mucus layer, and it can capture the virus before it even enters the host cell. Even if that fails, there are tissue-resident memory T cells and B cells that can be established in these mucosal organs to quickly eliminate infected cells before the virus has a chance to spread. And so this would result in reducing not only infection, but transmission as well as reducing the burden of disease within the infected individual, because we're stopping the virus from spreading right at the border. This is akin to putting a guard outside of the house in order to patrol for invaders, compared to putting the guards in the hallway of a building in the hope that they would capture the invader. So how do we elicit these mucosal immune responses? Well, most licensed vaccines that are used in humans are currently live attenuated pathogens, which cannot be used in immunocompromised individuals. However, we came up with a new strategy called Prime and Spike, which leverages the immune responses elicited by conventional vaccines, like the mRNA vaccine, and convert that into a mucosal immunity by spraying into the nose recombinant spike protein or mRNA encoding the spike protein that's encapsulated into immunosilent nanoparticles. Using this prime and spike strategy, we are able to inhibit infection, transmission, as well as disease burden in animal models. I'm a co-founder of a company called Xanadu Bio, which is trying to raise enough funds in order to take this to human clinical trial. However, we are not there yet. It's still very early stage. So I'm really here to talk about the need for the collaboration between the government, the private sector, and the academia in order to bring these innovative strategies forward as quickly as possible. And you will hear other creative strategies from the panelists here today, but it's very important to note that we need to move quickly to start testing these nasal vaccines in humans, and that requires a significant U.S. government input, both resource and help with manufacturing and delivery, as well as acceptance across the society. So I'm really happy to be here on the panel.
Thank you very much. Now let's hear about two of the different approaches to nasal vaccines. I'll begin with Dr. He. Please tell us about what CyanVac is doing.
Hello. My name is Biao, so in case you're intimidated by 3 vowels in my name, it's pronounced meow with a B, so don't forget B with meow with a B. So first I'd like to thank the organizers for the invitation. I'm really excited to be here to tell you about the progress we have been making to develop a human intranasal vaccine platform. I am Fred Davidson Chair of Veterinary Medicine at the University of Georgia College of Veterinary Medicine. And as well as Dr. Collins mentioned, I am the founder and CEO of CyanVac. And Dr. Fauci mentioned, and Dr. Iwasaki mentioned as well, the infection of many pathogens to human came through respiratory tract, which often means nose. And then we really need to find a way to stop the virus where it enters us. So for past 25 plus years, we have been developing a human intranasal vaccine system uses a nasal spray to deliver an antigen, to immunize people. So I look around, I suspect many of you have dogs at home. And if you have dogs, you most likely have your dogs vaccinated with the kennel cough vaccine. So kennel cough vaccine is given to your dogs through dripping the vaccine into the nose of your dogs. So in the dogs who have been using this vaccine for over 40-some years now. So in past 20-some years, so we have been essentially to re-engineer the component of this kennel cough vaccine. So we repurposed the virus in this kennel cough vaccine called the parainfluenza virus 5, PIV5, to use it as a human intranasal vaccine platform. So using this human intranasal vaccine platform, we have, of course, developed many vaccine for nasal inoculation. For instance, respiratory syncytial virus, and of course, most recently, we pivot to do COVID-19 virus vaccine. So the vaccine is right now in human clinical trial in the U.S. So we're very excited to be here and to tell you about what we do in this vaccine and this platform system. And last but not least, I really want to thank my collaborators and supporters over the years to make us go this far. One of the places I really want to thank you is NIH. So, it's as they mentioned, and I think Dr. Kessler and Fauci, Dr. Fauci mentioned, if you look at the slide, I'm very happy he put our company on one of the intranasal vaccine that's supported by NIH. Because over the years, when I started as an assistant professor, I had this idea. Back then, I didn't know our vaccine vector would really work for whatever we're doing now, right? We got funding from NIH. NIAID mostly supported us for over the years, so I'm really, really grateful. So I'm here, and I'm excited about what we're doing, and I'm very hopeful and very optimistic that our intranasal vaccine platform system, as well as our intranasal COVID-19 vaccine, will contribute to control of the COVID-19 virus infection. So I will stop here.
Thanks very much. Dr. Marty Moore, another representative of a company that's in the leading role here trying to develop intranasal vaccines at MISA. Tell us about your approach.
Thank you. Yes, my name is Marty Moore. I'm a PhD virologist, co-founder, and chief scientific officer of, it's pronounced Mesa Vaccines, which is a clinical vaccine biotech. So, the pandemic really exposed the vulnerability of America's health and way of life to viruses. As we heard today, fortunately, life-saving injectable vaccines were rapidly and successfully deployed, saving over 2 million lives. So why are we here talking about the need for new, different vaccines? Let me paint you a picture of two futures. The first future is the road we're on, where the vaccine is essentially transmitting and spreading unchecked. And this drives the evolution of new variants and outbreaks. The virus is mutating faster than our ability to manage vaccines, and not enough people are getting boosters. These injectable vaccines do a great job at preventing severe disease, but they do little to prevent infection and transmission is the engine of variant evolution. So in this future, right, COVID is a consists of rolling outbreaks of disease disruption, and the alternate future. So what's the alternate future? Well, the alternate future is based on a successful roadmap, the path we took with polio virus. Okay, the first polio vaccine, like the Operation Warp Speed of its day, was injectable, was rapidly developed. It worked and it saved lives. But that wasn't enough for polio, and it's not going to be enough for COVID-19, because vaccines that you administer with a needle don't prevent viruses like this from entering your body. To do that, you need mucosal immunity, as Akiko said. Okay? So, subsequently, the generation of a second-generation polio vaccine did this. It provided this frontline immunity, it reduced transmission, and this was the key to controlling polio globally. We can follow in these footsteps. We can develop transmission-blocking COVID-19 vaccines as an end-game strategy. How doable is this? What do we really know about mucosal immunity and COVID? Well, we're still learning. We know that SARS-CoV-2 infection itself can prevent reinfection. It actually does so better than injectable vaccines. We know that infection stimulates mucosal immune responses in your nose, and that natural infection can prevent reinfection from variants. How long does this natural immunity last? We're not certain, but the data point to 1 or 2 years, which is not bad. But this natural immunity comes at too high a cost, right? Disease. Our goal in developing nasal vaccine is to replace infection with a safe nasal vaccine that gives you similar or hopefully better mucosal immunity. I do think nasal vaccines can work better than natural immunity. At our company, Mesa, what we've done is we're developing a live viral intranasal COVID-19 vaccine that preclinically showed induction of mucosal immunity, systemic immunity, had high efficacy. We've enrolled approximately 100 subjects in a phase 1 clinical study, and thus far we're seeing great safety with no what's called vaccine shedding and promising immune responses. So we're poised for acceleration, and our technology has been studied for another virus and 3 other phase 1 clinical trials. Look, We know from 200 years of vaccinology, going back to Jenner's smallpox vaccine, that vaccines that mimic natural infection tend to work best. They can be broad, they can be durable, they can block transmission. Okay? So, this is our goal. But no one here today can tell you that mucosal COVID vaccines work. We're not there yet. We need clinical efficacy data to answer that question, and that's why we're here talking to you. So clinical efficacy plus immune correlate studies together can answer the questions, not just do these vaccines work, but how do they work? And that could not only potentially deliver this knockout blow to COVID, a transmission-blocking vaccine, but also provide valuable information about vaccines to fight this virus and future pandemics.
Thanks very much. Now, for something completely different, let's move from the nose to a new way to utilize the skin. And let's hear from the CSO of Micron, Mark Prausnitz, who will tell us about microneedles.
Well, I'd like to tell you about a different delivery technology, a technology that looks like this, a small patch that can be applied to the skin, a simple press, leave it in place a few minutes, peel it off, and discard it. We think that this can not only make vaccines more readily accessible, but also improve their immunogenicity. So I'm Mark Prausnitz. I'm representing Micron Biomedical today based on technology that was developed with support from NIH, NSF, CDC, Gates Foundation, and others at Georgia Tech, and has now been commercialized for the last few years via Micron Biomedical. So this patch targets vaccine delivery to the skin. The skin is another surface on our body. The body is equipped to mount immune responses especially well to pathogens that appear on the surfaces, because that's, of course, where they first appear. So what we and others have found is that if you vaccinate in the skin, you can get a more robust immune response as compared to vaccinating deeper in the body, such as in the muscle. Indeed, the only human disease that has been eradicated, smallpox, was done through vaccination in the skin. So in addition to that example, in the studies that have been done with other vaccines, what we have seen is longer-lasting immune responses, broader immune responses, stronger immune responses, dose-sparing enabled by skin vaccination. And for these reasons, we're optimistic that skin vaccination can offer a number of immunological advantages. But what also drives this are the logistical advantages. You saw how simple it was to apply one of these patches. In fact, in the clinical trial we did on flu vaccination, we included a group where self-administration by the subjects was part of the study. They saw a few PowerPoint slides, they applied the patches, the results were just as good as when a nurse applied those patches. So we do envision a future where certainly administration by minimally trained personnel could be done, of a special value in rural settings and in developing country settings, and potentially even self-administration when that's appropriate. These microneedles are as long as a piece of paper is thick, and so as a result, you barely feel them, they don't hurt, you don't see them, they don't look scary, and so as a result, acceptability of the vaccination should be higher. When the needles enter the skin, they contact the fluids inside the body where they dissolve. So you get the added benefit of a dry vaccine that is more thermostable, and we've shown for some vaccines that we can store them for 2 years at 40 degrees Celsius, so as hot as it gets in London a few days ago, and they're able to maintain that stability for years. But you don't have to redissolve it before you inject it. You take the dry formulation, you stick it on the skin, and it dissolves away in the skin, leaving no sharps waste, so that sharps waste disposal is no longer an issue. Manufacturing can also be done at a cost that's comparable to a pre-filled syringe. So we're optimistic about the future of vaccination that can be more accessible to people as well as be more immunogenic. Thanks.
Thanks very much. Now let's hear from Dr. Michelle Smith from the Arkansas Department of Health. Is this all going to make sense to people who are looking for a way to prevent illness?
Well, I hope so, and that's one of the reasons why I'm here. I'm also very excited to be on this panel. Thank you to the organizers. Because a public health professional, our job is to learn this information digest it, and then put it out to the masses so that they can understand all of this fancy jargon and patches and everything. So as a reminder to public health professionals, I just want to encourage everyone to remember to always put public trust, trying to build public trust at the forefront of your work, because no matter how groundbreaking, how effective, how cool these patches are, if they aren't utilized, if people don't have access to them, then they won't do what they're designed to do, and that is to save lives and protect the economy. So how can we work from moving your advances from the lab and bringing them into American homes? First, I'm going to talk briefly about what Arkansas has been doing in terms of vaccine delivery, and then I want to highlight 3 Arkansans and highlight how these advances could help them. So first, to offer some context about Arkansas, our population is mostly rural. We have about 3 million people. We have high rate of comorbidities such as obesity, diabetes, cardiovascular disease. And before COVID, our office worked with non-traditional partners such as barbers, beauticians, fraternities, sororities, HBCUs, and faith-based network. So when COVID did come along, we were already well established in these networks, and so they could trust us already. And so we were able to quickly mobilize them to help us. They were like our boots on the ground. We would give them factual information about vaccines, about COVID, wearing masks, and we had town halls, webinars, fact sheets. And so it was great to already have that trust built into our network, so then they could take that information and then spread it out to individuals in their network. One example of how we used a health equity lens to vaccinate or ensure that vaccinations were accessible in minority populations is that we listened to a sorority when they called us and said that they had been calling the pharmacies and weren't able to get on a waiting list, much less make an appointment. So we listened to that. Based on that concern, we reached out to the Arkansas Pharmacists Association, and we asked them to open more slots for minorities in their communities. And once that worked, we said we need to expand this, and so we developed health equity strike teams, where we would go to communities, minority communities, and bring the vaccinations to them. We had teams in low vaccination counties, and we used lay people, we used pastors as volunteers, because again, that trust, they knew someone, they were familiar with them, and that encouraged them to come get tested. Partnering with the Arkansas Pharmacists Association also gave us access to pharmacies that had these sub-zero refrigerators. A lot of the small pharmacies did not, and that's one of the advances that I'm excited about when you don't have to have that sub-zero temperature. This will allow us to go into more rural communities and vaccinate without having to have a network, which was good. We did have a network where the pharmacies would share with the others that didn't have that. But if they don't have to have that barrier, then we can vaccinate more individuals. So as a result of our health equity strike teams is what we call it, we had 127 vaccination clinics, and just from our team alone, we vaccinated over 19,000 minorities. Now, if I can, I want to just put a face to some of these numbers. I want to talk about some individuals and how they could have benefited from these things. First, I want to talk about Rosie Lucas. She was a community member she actually helped us when we first started doing our testing. She called people, made sure they came out to get tested. Well, Rosie, who happens to be my aunt, she was admitted to the hospital because she did not want to get vaccinated. She was, a little leery of the needle. So some of these nasal injections, this would definitely help her. And this impacted me because, of course, that was my family member. But I'm getting the eye over here, so maybe I can go back and talk about some of the other Arkansans that can benefit from this. But I want to end with a note to everyone up here to remember not only the adults but the younger generation. I've come in contact with some kids that will blow your mind. And because they're watching the news, these types of things excite them. But we don't have a lot of diversity. And so I think that's another way to build trust. When we have 20 years from now another panel like this, and we have African Americans, Latinos up here, then they can go into their network and talk about these advances, and then we'll have greater access to these technologies.
Great, thank you very much. Sorry for giving you the eye. So let's get into some questions here that I might throw at my panelists without having warned them, but these aren't too bad. One thing I think when it comes to the use of nasal immunization, the one that people are familiar with in terms of a human application is for influenza, and it's flu mist. And there's a general sense that it was okay, but maybe it wasn't quite as good as people had hoped. What did we learn from that? And is there some lesson there in terms of how you would apply this for COVID? Maybe I'll ask Dr. He to start that response.
That's a really good question, because in animal, in veterinarian field, we've been using kennel cough vaccine for over 40 years now. It's been used really well, and there's great acceptance of the kennel cough vaccine, and they continue to be a vaccine that's been used. And for influenza vaccine, and I'm a big fan of flu mist, so some of you may have gotten it, and it's really a wonderful vaccine. And I think the lessons for me from scientific point of view to learn was that it went through a lot of difficulties in developing it. It's not because it's not a good vaccine. It's a wonderful vaccine, and you just get a spray in the nose. You don't have to get an injection, right? How easy is that? But the issue, when you do something new, when you do something novel, right, it's exciting, but it's hard. Innovation is hard. I want to point this out. We have been doing this, as pointed out, for 20-some years now for intranasal vaccine, right? So the lessons we learned, and as in the early panel, we talked about what is immunocorrelate, right? In the case of influenza virus for flu mist and MedImmune, which you later acquired by AstraZeneca, I know those people who worked on the project. They were fantastic, scientists. And yet, when they do this, they have to deal with what is the immunocorrelate you can use, because it's a new vaccine. So traditional flu vaccine, we have an immunocorrelate. It's called HAI titer. And then you do that, you can basically very quickly get the approval. So when you do something new and innovative, and then what would be the immunocorrelate? Because this time you're using a live virus. And can you look at the IgA, or can you look at the the cellular immune response? All those kind of things which are critical for the efficacious effectiveness of flu mist, but there is no standard from FDA. So this is one of those things I hope when we move forward with the COVID vaccines for intranasal. I'm sure Marty was thinking about the same thing, right? and early on, what would be the criteria, right? What would be the immunocorrelate we're going to use to move this forward to get approval, the BLA?
So you've raised it. Let me ask Marty, what would you say from your perspective would be most helpful here?
Sure. Let me back up and say, when flu mist was at its peak, it wasn't just an approved vaccine. It had a preferential recommendation in children over the flu shot. And the reason for that preferential recommendation is that it had better efficacy. And if you look at the data, what can we learn from flu mist? Well, it was more durable. And in the so-called school mist studies, it showed that it could reduce transmission. So these are the principles of mucosal immunization that we're aiming for. Now, what happened with flu mist? Obviously, it fell off, and I think without getting into the weeds, these are influenza-specific and manufacturing issues. But at its peak, when it was made correctly, it was an outstanding vaccine, especially in children who are more seronegative to influenza.
Thanks. Helpful to have that clarified.
But the correlative protection question is a good one. So, this additional efficacy in children that we saw with flu mist did not correlate with the HAI titer, right? So there's something else, and that's when you're using a live viral vaccine, especially if it's mucosal, there's a double-edged sword. You're stimulating a broad immune response, innate, adaptive, cellular. You're not hanging everything on a serum antibody response. So it's more difficult to measure these mucosal immune responses. And I think Dr. Fauci brought that up in his initial presentation, that we need to better understand mucosal immunity. Well, we also just need more standard assays to study it, among us.
Yes.
So I think we could make some progress there.
And that was on his list that I quickly copied down of 7 barriers for making progress in mucosal application of vaccines. Let me ask Dr. Iwasaki, one of the things that I think you've written about is how it seems to work particularly well if you have a standard injectable vaccine and then you boost with a nasal vaccine. Can you say something about that? And for the case of COVID-19, where we hope a lot of people will have gotten that initial vaccine, is there something special there that we should pay attention to?
Yes, thank you very much for that question. Just to kind of round out the flu mist question, one of the issues about the flu mist and other sort of live attenuated pathogens is that once you develop enough antibody against the actual vaccine, the immunogenicity declines over time. So that's one issue that we need to keep in mind, that if we're going to be boosting over and over through the nasal route, we should be really paying attention to what kind of platform we're going to be using over the long run. And just to come back to Dr. Francis Collins' question, indeed our approach is actually leveraging the pre-existing immunity that's generated by the intramuscular vaccines. And how it works is that you generate these memory T-cell response that's circulating, and we leverage those T-cell memory to serve as natural adjuvant. So most vaccines require both antigen and adjuvant. Antigen is the target that B cells and T cells see, and the adjuvant is the molecule that stimulates the innate immune response. Without the adjuvant, it's very difficult to engage adaptive immune response. However, what we found is that these memory T cells can serve the function of adjuvant altogether, and not requiring any adjuvant to be delivered as a nasal spray when we do the prime and spike approach. And the other advantage, I guess, would be that because we can use the mRNA that's conjugated, complexed into a nanoparticle, that platform will not suffer from pre-existing immunity. There's no antibody against the nanoparticle or the mRNA itself to block that from becoming effective. So there are a few advantages. Every platform that we have here have pros and cons, and I'm just raising some of them.
That's really helpful. Let's talk again about microneedles. So Dr. Prausnitz, it sounds like there's a real promise here. I'm not sure I got a clear sense, though, about how this compares with intramuscular injection or nasal vaccination, if what you're trying to do, which is really important right now for COVID-19, is to prevent transmission. Do we have any way of guessing how that works for microneedles applied to the skin? What does that do for your mucosal immunity?
There have been studies comparing microneedle patch vaccination with the intramuscular vaccination, and those have been favorable. By some immunological measures, the responses are similar, and by other measures, you get a superior response when you vaccinate in the skin with the microneedle patch compared to intramuscular. I'm not aware of any comparisons between the microneedle patch and, say, nasal or wet mucosal vaccination. Indeed, the skin and the nasal mucosa have a similarity in that they're both surfaces and can leverage the immune system that is heightened on those surfaces. But the skin is not the nose, and if nasal transmission or pulmonary transmission is the mechanism, then the skin is not the site of entry for that virus. So it's unclear what role the microneedle patch might play in IgA or mucosal responses, but in terms of the systemic IgG responses, they are strong and favorable compared to intramuscular.
And let me ask about manufacturing. Seeing the microscopic pictures of these microneedle patches are pretty amazing. Is this something that is challenging to set up as far as a fabrication facility, or is this like no big deal?
Somewhere in between. So the way that we make them is we have a mold, we pour a solution on, let it dry, we pour another solution on, let it dry, and put it in a package. So the manufacturing process in principle is a simple one. It's one that pharma knows how to pour films and let them dry. There is the devil in the details. The manufacturing infrastructure does not exist today for mass production. It's under development. It should be available in the next few years, but the expectation is that manufacturing can be done at low cost, at costs similar or maybe even less than a pre-filled syringe. but that manufacturing infrastructure does not, in fact, exist yet today.
One of those challenges on Tony's list, I think. Dr. Smith, I cut you off, but I think I want to hear another story or two about how this set of new technologies might be well received or not in populations that maybe have not been particularly well served by our healthcare system and maybe even are a little less trusting that the next thing that comes along is actually good for them. So say a bit more about that.
I really think that preventing transmission holds the most promise. Unfortunately, breakthrough transmissions are causing people to be complacent and just kind of lose hope in everything and just say, you know, what's the point? I'm still going to get infected. There was one individual, his name was Dale Smith. He first was hesitant to get vaccinated, but his wife got vaccinated, and then he did. And he became a very strong advocate on social media. And he used that platform to kind of convince his friends, and he was a musician, other musicians, to get vaccinated, use masks. Unfortunately, Dale became ill, was hospitalized, and because of his one of his comorbidities, he died 5 days after being hospitalized. And so that gave a lot of ammunition to the naysayers that were already saying, you see, I told you I did, what's the point? So I really think that that will help people once they see that this particular vaccine can stop transmission. I think that holds the most promise. But I also want to stress the importance of talking to individuals about what to do after they are infected. A lot of people in the minority community don't know about the treatments. We spend a lot of time talking about the science,